Welcome everyone to the NGSeminar, and thank you very much for all your patience. Today we have Dr. Andrey Prjibelski, who will introduce us to long-read RNA sequencing. And Andrey graduated from the St. Petersburg Academic University in Russia with a master's in software engineering and informatics. And currently he is employed as a research fellow in the Center of Algorithmic Biotechnology, St. Petersburg University. And recently, in 2020, he defended his PhD thesis entitled Algorithms for Genome and Transcriptome Assembly. His current interests include transcriptomics, genomics, and sequencing data analysis in its different forms. We invite everyone to ask questions for Andrey during the seminar in the chat section. And for those on Discord, it will be on the Isoforms channel. So thank you again, Andrey, for waiting, and the floor is yours.
Thanks a lot. If anything is unclear regarding, content quality or anything is unclear about algorithms, just let me know, and we will stop there because I think we have plenty much time, and we're not in a rush at all. Can you still see the video? No?
It seems loading for me.
Second. You see it now?
Yes, I see it now.
Okay, great. First, the outline. In the beginning, I will talk a bit about long-read RNA sequencing in general, like what kind of technologies we have here, how do we align those reads, and what are existing solutions to this kind of data analysis. And then the main algorithmic part will come. I will talk about what we did in our lab, how we address the problem of analyzing long-read RNA data, and in the end, some results and discussions probably. Long-read RNA sequencing is not very different from what you have in long-read DNA sequencing, except DNA long reads have been used already for almost 10 years, maybe a bit less. RNA long-read sequencing appeared rather recently, like maybe 3 years ago or so. And of course, we have PacBio as a pioneer of long-read sequencing there. We have Isoseq protocol, which uses typical PacBio sequencing machines with some additional library preparation steps. We have usual PacBio, like you simply apply almost the same version of protocols as for DNA sequencing. And we have, of course, PacBio CCS, which are now called hi-fi reads, which are very high quality reads. And PacBio CCS, or now how they call them now, hi-fi reads, they're obtained through multiple sequencing of the same fragment. You make a circular template, and then you sequence it with usual PacBio several times. Of course, as we know, PacBio has very high error rate, so you get a lot of errors in each fragment. But since in PacBio errors are distributed randomly and uniformly, there are no biases like homopolymers and stuff. You get random errors, and just using consensus, you can correct them and obtain very high quality read. And as they claim, the error rate is even lower than in Illumina reads. And genomic data, you usually want the length. You prefer length over accuracy. In RNA, you don't need like 50 kb reads. Like 5 or even less will be more than enough to sequence almost every mRNA in your sample. So hi-fi reads is exactly what you need for RNA sequencing. And of course, nanopore reads, they have two different protocols, cDNA sequencing and direct RNA. Direct RNA is a bit more complex from biotechnological side, as far as I know. They don't have anything like CCS reads. Everything is raw nanopore, so these kind of data you always get higher rate, but of course they're still much cheaper than PacBio. And the first thing you want to do with those reads is you want to map them onto the genome, and there are some aligners that can do that. The first long read spliced aligner was GMAP, as far as I know. Later we got STAR, which has a special model called STAR-long for long read sequencing alliances. But both of those, both GMAP and STAR, they are not designed for long read sequencing. They're designed for correct transcript sequencing with very low error rate. So they can be applied for PacBio HiFi, but not for raw PacBio reads or to nanopores. The first mapper that was able to map long reads with splicing and considers higher rate is Minimap2, and I think it does it pretty well, and of course, as you know, at a very high speed. Recently, there was another aligner called DSALT. People claim that the quality of mapping long RNA reads to the genomic sequencing by DSALT is better than for Minimap, but it's very slow, especially for index construction. And what prevents us from mapping all those transcripts exactly to the reference genome? Like, you probably imagine that if we have a correct transcript sequence, like obtained from a sequencing machine, then the problem is solved and you have nothing to do. Well, in a perfect world, it is so. Reality is more harsh because the isoforms of multiple genes, they have very complex structure in mouse and human genomes, and usually currently scientists work with those organisms the most. So you may have, for example, short exons like we have here. The green is known isoform structure, and yellow is our read. And the exon is so short that, for example, if you use minimizers or just k-mer matching as a base for your aligner, there is not enough nucleotides in this short exon to be matched exactly, and you just skip it. Of course, you may have also short introns, which you may confuse with the insertion or deletion. And I have similar exons. So I even saw isoforms that have different structure according to the annotation, but there are two exons of exactly the same length and exactly the same nucleotide content. Of course, when you do mapping, you cannot consider which one is correct because they're absolutely the same. And there are a lot of exons that have very similar nucleotide content. And of course, if we talk about raw PacBio sequencing on nanopore sequencing, higher rate is also what complicates this problem. You have fuzzy junction sites. That means you cannot exactly detect where your intron is because of errors near the splice sites. And again, because of poor read quality, you may not map short exons, even though there may be like not 10 base pairs, maybe more. But if you have 10 errors in 50 base pair exon, it's very likely that you will not map it correctly. And one thing I would like to mention here is that all spliced aligners that I'm aware of, they tend to map reads or transcript sequencing so that you meet that introns have canonical splice sites. As you may know, we have 3 different splice sites which are called canonical. 4 of them are processed by major spliceosome, and 1 is processed by minor. And GTAG is more than 99% of splice sites in human and mouse. So when Minimap or Gmap or Starlong tries to map a sequence, it always considers that Intron should start with GT or AT, GC, and it should end with AG or AC. So as you see, this is a correct mapping, which we expect from our read. However, the aligner thinks that this kind of mapping has higher score. As you may see, it has smaller number of mismatches, and it also meets the criteria of canonical splice sites, GTAG. And of course, it's very easy to meet a random canonical splice sites near your correct splice junctions. So it's not very rare when a aligner correctly maps your Intron or skips some exons due to trying to make Intron fit into those canonical splice sites. Okay, any questions about aligners? Okay, if there are none, then we'll just briefly talk about what we have now about the tools which were developed for long read analysis. One is called CDNA Cupcake. It's like a bunch of scripts designed for read clusterization and collapsing. So the goal is to find reads that belong to the same isoform and collapse them into correct transcript sequencing, basically finding a consensus. More complicated tools are designed for transcript model constructions, Flare, Talon, and Bamboo. As you see, the last one is not even published. The first two are published during this year, meaning that they are very recent. And that proves the point that the field of long-read RNA sequencing is rather new, and the tools are just started to appear. And another category is reference-based assembly. StringTie2 is the modified version of StringTie, and Scallop-LR is the modified version of Scallop assembler. For me, actually, second and third category, transcript model construction tools and reference-based assembly, are very close, and I don't think you can really say what's the difference, because their goal is they take BAM file as an input of aligned long reads, they take existing annotation, and they try to create a new annotation based on your BAM file and gene code FF file. The only thing that this group of tools usually tries to believes more to the reference annotation and tries to find modification of existing isoforms and so on, while reference-based assemblers, they work without annotation at all, and they just believe reads more and try to construct some kind of size graphs and find out what the BAM file tells them. And another tool is, I think it's one of the most known tools in the area. It's Scanty, and they have different versions. Scanty2 is a forked version of Scanty1, developed by some other guys. Scanty3 is developed by the same guys as Scanty1, so it's a bit messy. But the goal of this tool is to classify different transcripts and detect various events, for example, intron retentions, alternative donor acceptor sites, and so on and so on. So our goal was to do something close to second and fourth category, construct transcript models, and find out something like what is the difference between what we see in reads and known annotation. First, the pipeline overview. As most of those tools, we take gene annotation as an input and BAM file with long read alignments. We prefer to use Minimap now, since it's the fastest one and works well with higher rate. Then we try to match every read against known isoforms from the annotation and say, okay, this read belongs to this isoform, this read belongs to another isoform, but with some modification. So we detect also inconsistencies between reads and isoforms. For example, we find reads with intron retentions and so on. And of course, we do quantification, which is very important in this task. because when you have short read data, you usually estimate your abundances using coverage depth or like TPM values. Here, you just, if you have a long read covering some known transcripts, you can be sure that, yes, you had this molecule in your sample, and you can be sure that like the expression values are much more certain, and they are more reliable in terms of how they represent the original molecule quantity in your sample. Okay, so how do we map reads to known isoforms? Let's look at the simple example. We have 3 different isoforms of the same gene: the green, orange, and purple one. Maybe the colors may vary at your screen due to video compression, I don't know. So what we do first is we take a look at all annotated introns, and we enumerate them. Here's the intron 1. It appears in all 3 isoforms. Intron 2 appears only in first isoform. Intron 3 appears only in yellow one, and so on and so on. So we go over the annotation. We enumerate all known introns according to the coordinate. And now we can say that each isoform can be represented as a list of numbers. And the number is like which intron is included in this isoform. So for example, green isoform can be represented as 1, 2, 5, 7, 9, and so on and so on. And to make it even more convenient, we actually convert it to a vector of a fixed size. The size is the number of introns. And we say put 1 if this intron is included in our isoform, and we set -1 if to the position where we say the intron is absent. So for example, for green isoform, we have intron 1, so we have 1 here at the position number 1. Second one is also included, so we have 1 at position number 2. Then we have -1 because intron number 3 does not belong to the green isoform. Intron number 4 also is not in the green isoform, so again -1, and so on and so on. So binary system, like yes or no, whether we have the intron in our isoform or not. I'll repeat that, or everything's clear? I see someone is typing question. Why do I use -1 instead of 0? Yeah, I had this question previously, a good one. We will use 0 further for another notation. 0 will mean we don't know. I'll explain it like a few slides later. Oh, and the next thing, the thing is called intron profile. Then we do the same. It's called exon profile. We split exons into non-overlapping regions. Of course, we do the minimal split. So, like for example, you see here we have a green small exon shorter than those two, within the yellow and purple. So we split it into 2 regions. And then we do the same thing. We see which exons contribute to which isoforms, and similarly construct exon profiles. Again, one says that this particular region contributes to the isoform, and -1 if it doesn't. Okay, now we have this exon regions and list of introns, and suppose we have some read which we don't know. We don't know which isoform it belongs to. We see what exonic regions are covered by this read, and we also see what introns belong to this read. By that I mean which splice junctions exactly correspond to known annotated introns. As you see, for this yellow read, we see that it partially covers intron region 3 and 8 and fully covers intron region 5. So we say, yes, we see 5 and 8. And splice junctions in the read match exactly introns number 3 and number 6. We construct here a vector, which is called read profile. And yes, here we use 0. For example, introns. We don't know whether intron number 1 is included in this read because read is truncated and it only starts here. So we put 0 here because we cannot say, was intron 1 within this read or no? Same about the exons. We cannot say anything about exons 1 and 2. We put 0. We can say definitely about 4, for example, because we see that the read spans it but doesn't cover. So we put -1 here. Similar again, exons 6 and 7, we put here -1s because we are sure they are not covered by the read, but not about 8. And the same for introns. If the intron is covered, not matched, then we put -1. If it's spanned and matched, we put +1. If it's outside the region, we say 0. We don't know. And then we have read profiles, and we have profiles for known isoforms, and it's very easy to match them since it's simply a vector of 3 possible numbers. You can just calculate, I don't know, Hamming distance and say, yeah, if we have a perfect match, then this read definitely belongs to this isoform. And simply matching to read profiles to isoform profiles we constructed previously, we can say that this brownish read belongs to orange isoform. And if you look at the structure, it really does. So at this point, you may ask a reasonable question: why do we do such complex thing as like constructing profiles, then like doing Hamming distance? You can represent your introns simply as a pair of coordinates, then match a list of 1 coordinate pairs versus another. And it seems much more than like straightforward solution. And here's the answer why. I previously told already that when you map your long reads to the reference genome, you may not exactly get your splice junction correctly. So, for example, you may have a read something like this one. On top, you have a read. It looks like it belongs to was originated from orange isoforms. But when it's mapped to the genome, you see all the splice junctions are fuzzy and are not exactly matched. So when we match read introns against known annotated introns, we allow a small delta. By default, it's 6 base pairs. If you have more error rate, you can increase your delta. If you have very accurate reads, you can decrease it to like 3 or even 0 if you are completely sure about your data. So when we match those, when we say that read contains an intron, we put +1 not only when we have exact match, but even when every splice site is located within 6 base pairs from the annotated splice site. So this the key thing of how we address the problem of the splice junctions is allowing inexact matches. So yeah, no rocket science here actually, but as I see, it's a pretty simple solution. Once you do it, you construct the same profiles and you match it exactly against known profiles of your isoforms. Also, what becomes simple when you use those profiles, there are a lot of different alignment artifacts beside the fuzzy splice junctions. As I mentioned already, one is exon skipping, when you have a short exons and you just miss it when you align your read. And another one is intron shift, I also mentioned it, when your aligner for some reason or due to higher rate decides that your splice junctions should be located differently. For example, below you have the annotated isoforms and on top you have read, and you see the intron is shifted to the left because part of second exon is aligned to the beginning of third exon, as I showed previously. So once you have your profiles, it's very easy to detect those kinds of artifacts, even when the difference between splice sites is higher than delta. So you don't have this match, even inexact. You can see, okay, yeah, we have matched introns number 1 and the last one, but the middle one is not matched. And we can see that the difference is small and then say, yeah, this read is probably originated from this isoform, but there is an alignment artifact. And it's also easy to report not only alignment artifacts, but some real modifications such as Intron retentions. You just simply see that your annotated isoform contains 1 there, and there is nothing. There is -1 in your read profile. You can also detect mutually exclusive exons, alternative and acceptor and donor sites, so alternative introns, exon gain and exon loss, and so on and so on. So all kind of different modifications you can imagine in your RNA molecules. Okay, if everything is clear, I'm ready to move to the second part. So yeah, the questions. Will very short introns exons be problematic? It makes sense to take into account intron exons, aligned length, as well as presence, absence. Very short introns, and especially exons, are super problematic. So when we do this comparison, when we try to match a read to isoform, we actually do consider the length. That's a very good point. So, for example, if we see exon of length, I don't know, 300 base pairs skipped, we say, no, this is something wrong. This is probably not correct. It just cannot be a simple artifact alignment. Probably it's modified RNA. Probably it's the difference in splicing. But when you see, I don't know, 20 base pair exon missed, you say, yeah, probably most likely is due to alignment artifacts. And what we try to do now is we aim to verify the alignment. So, for example, yeah, let's go back for since. For example, for example, like here, you have a green exon skipped due to errors or just because it's very short. And you have like, but the read still contains some parts of these exons, even with errors. So aligner has to map them somewhere. It cannot just throw away the sequence and say, I don't know where to map it. It tries to attach them to the previous and the next exon. And so you have incorrect splice junctions here also. So what we can do is we can take those red parts, which are presumably incorrectly aligned, because we know the correct annotated splice junctions here. We can see, are they anywhere close to, like, is there a sequence close, like, to what we see in the annotation to this short exon? And if the alignment is more or less good, then we say, yeah, this is minimap error. It should be like those red parts should be aligned here. If it's very different, for example, the total length of those red parts is not equal to the length of the exon, it's like very different, then we say, yeah, something is wrong with the original molecule, not the alignment. Yeah, good point by Maya. We do consider the length of the exons and introns when matching isoforms to the annotation. Okay, and the second part, of course, people are like read classification is important. Yeah, you can say, yeah, this read belongs to this isoform and this one belongs to another. But usually people want to see transcripts that express in their sample, either the expression or, like, since you have the long reads, they also want to see novel transcript models. So what do we do then? We have every read, not every read, but most of the reads assigned to some isoforms. Of course, in case when read is very much like truncated heavily, you cannot say which isoform is. You just drop the read and say, yeah, we don't know which one is it. For example, you can say that the read is ambiguous and can be fully well matched to different isoforms. You have always something like DNA contamination and reads that map into intergenic space. You usually ignore them. So in the end, we have reads that are matched to the isoforms consistently without any modifications, and inconsistent splice matches. Consistent splice matches is that don't have any Intron retentions or alternative splice sites. You just say that, yeah, this read belongs to known transcripts, and you simply quantify them and output known transcript directly from your annotation to your output file. From inconsistent splice matches, by them I mean reads that have some differences in comparison with annotation, we try to group the reads together which have the same modification and attempt to construct a novel transcript model and output them also in GFF file. So how do we do with that? Today we have the same example, the same gene with 3 different isoforms, and let's say we have a lot of reads mapped to this gene, and they look like this. And first, what we do is we group reads according to isoforms they belong to, according to our assignment, and also we group them by modification type. For example, brownish reads belong to orange isoforms, but with Intron retention. They fall into one group. Reads that would be aligned to simply to orange isoforms without any modification would fall into consistent group and will not be included here. Those orange or reddish reads, they belong also to orange isoform, but with alternative donor site. As you see, second exon is modified. Those reads still look more similar to orange isoform. And another one, for example, is group of green reads which have a exon loss, skipped exon in the end. So we process each of this group independently. Take a group, for example, we took all the green reads that belong to green isoform and all the reads that have these exons skipped. And then we select some representative read, which has, for example, the highest coverage, probably should be the second one or the last one. Doesn't really matter in this example. And we take this read and we try to basically merge it with annotated isoform. So construct something like a new isoform. And we have a matched splice site, matched Intron, for example, like first, second, third. There may be fuzzy splice junctions here, but we still consider this as a matched Intron. We take exon and introns from the annotation, correcting fuzzy splice junctions. But when we have a detected modification, not like alignment artifacts, but a true modification, like skipped long exon here, we take it from the read. So this is some kind of using read and annotated isoform. And what we get in the end is some bluish isoform with which is a copy of green isoform, but with the exon skipped. Then we constructed that new transcript model, and we go back to our group of green reads, and we verify whether those reads do really support this new isoform or not. If this new isoform has high support, I don't know, depends on your settings, like, I don't know, 4 or more reads by default, say, yeah, we will output this isoform in GFF file because we really trust it. And a more exotic situation when you have reads grouped, they belong to the same isoform, this reddish and yellow or brown reads. They all belong to orange isoform, and they all have the same modification, alternative donor site at exon number 3, or Intron number 3, as you wish. But as you see, some reads support one splice junction, and other reads support another splice junction. So how do we proceed in this case? And I'm not sure it's very often. It happens very often, but it may happen. Why not? Again, we select a representative read that has the best coverage. For example, this one. Again, merge this read with own isoform, correcting only the modification side. This one. So we have a copy of your orange isoform, but with another modified exon. Then we go back to this read group and see which reads do support our novel isoform. And it appears that none of them support it. Only first 3 reads support this modification, and the rest, which I colored gray, do not. So we throw away first 3 reads. We say, yeah, those reads showed us that they belong to this novel isoform. We don't need them anymore. We consider only the rest of the group, which supported neither the original isoform nor the new one. Again, we take this group, we select the representative read, we merge it with the annotated isoform, and go back to this group and see which reads support this second novel isoform, and in this case, it appears that they all do. So we do the process iteratively. We pick a read, construct a novel isoform, we throw away the reads that support this novel isoforms, and we do it until our group is empty. So for this group, we construct 2 different isoforms. Yeah, any questions regarding that? Okay, in any case, you will have a chance to ask any questions in the end or even later by email. Another problematic things are transcript ends, since I already mentioned that reads can be truncated, and it applies mostly to nanopore reads, since when you use PacBio CCS, you usually know whether you have a complete read, because you can see the adapters and so on. In nanopores, you often have a truncated version of your mRNA molecule, so it's not very easy to understand where you should end the transcript model, whether you should use the annotated transcription end site, or your reads are truncated because there's really another isoform that has different transcription end site. So here, what you can do is you can try to find poly-A tails. As you know, mRNAs are polyadenylated in the cell, and if a read is not truncated on 3' end, then you may find their poly-A site, and if you find them poly-A sites in multiple reads at the same time, you can say, yeah, this isoform really ends here. And if you see that your poly-A tails are not where your annotated isoform ends, you can report a different transcription end site, and it's usually called alternative polyadenylation site. But of course, for this type of event report, you need multiple reads, so having poly-A tails and ending at the same position, or like nearly the same position. And the same thing about 5' ends. It's not very clear how to detect transcription start sites, since you don't have any poly-A's there. Solution here is a bit more complex. As far as I know, the most common solution is to use additional data, which is called cage-seq. It's some kind of 5' sequencing, where you get reads only from 5' end of mRNAs, and then you can basically find peaks where you have a lot of where your reads start. And we have a cage peak there. It means that some transcription is definitely starts there. Of course, you cannot be sure which isoform is it, but if you see a cage peak near your presumable molecule start, and you see a lot of long reads starting at the same position and a cage peak there, then most likely you are right, and this is the real transcription start site. And yeah, you can report it in your novel transcript model. But this requires additional sequencing experiments, which means time and money and additional data processing. But for large experiments, probably it's worth it. Okay, more or less it about the algorithm. Time to ask more questions. Okay, let's move on to results. Probably this will be not that interesting, but of course I have to somehow justify that what we did, it makes sense. Again, our pipeline consists of 2 different steps: read assignment, and second is transcript model construction. So we did 2 different types of experiments with our tool. First one is we wanted to check how well we assign reads to known isoforms. And of course, when you sequence real data set, you never know what's inside your sample, so you cannot really say whether is your algorithm correct or not. So we use simulated data. There are currently 2 tools which allow to simulate long-read RNA data. First one is super simple. It's IsoSec-Sim. It's never been published. Seems like some student's project. So basically what it does, it takes a known transcript, truncates it on the both sides according to distribution you provide, and then it's randomly and uniformly inserts errors just by going over the read and like flipping a coin each position. Yeah, there is a question. Do you preserve? The question is, do we preserve poly A tails? To best of my knowledge, IsoSec 3 clips poly A tails sequences prior to FLNC construction. Yeah, that's right. We aim at processing raw reads with poly A tails. So if you would like to use reads pre-processed with IsoSec, which really does truncate all adapters and poly A tails, we now aim to implement a simple functionality which will take as input also IsoSec output. As far as I know, IsoSec tool outputs reads without poly A tails and adapters, and also some kind of meta information about every read, like whether poly A tail was found, whether adapter was found, and so on and so on. So instead of finding poly A tails ourselves, we will be using poly A tails reported by IsoSec tool. So currently it's not implemented, but as far as I see, it's not a big deal to just parse IsoSec output and add information for every read. Yeah, but a very good comment. And at some point previously, I was not to say surprised, but like I opened the file and I realized that there are no poly A tails in PacBio processed reads. Okay, sorry for a bit messy, but the question came to Discord a bit later. So back to simulation. Again, IsoSec-Sim is a tool which does simple simulation of PacBio reads and errors are distributed uniformly, which is actually very close to what PacBio has. Maybe not exactly repeats PacBio hi-fi pattern, but raw PacBio reads do have uniform error distribution as far as I know. So, and another tool is TransNanoSim. It's more sophisticated. It learns a model on real data and then tries to insert some nanopore specific errors. And also it can do Intron retentions and introduce junk reads and so on and so on. So reads produced by TransNanoSim are more close to like what nanopore sequencers do produce. And to test our tool, we simulated several types of data. We used reference transcripts, so full-length transcripts with 0% error rate. We simulated PacBio CCS with about 1.5% error rate. Probably it's a bit more than they have, than they report on their website. They say like their error rate is very low, but it all depends on how many consensus passes, like how many raw reads you use for your consensus. So 1.5% is obtained from our real PacBio CCS data. And we also simulated 2 different nanopore data sets, one with 11% of uniform errors. So it's not real nanopores, it's like very erroneous, like closer to something to PacBio raw reads. And we simulated nanopores with trans-nano seem to get more ONT-specific data, but with the same error rate. So this is a bit more detailed information about how we simulated the data, the mismatch rate, insertion rate, and deletion rate. All rates were obtained from real data. So, and also we simulated exactly 10 reads for every annotated isoform. Why is that? Because, for example, if you do something with more realistic coverage, for example, you have, I don't know, 10,000 coverage for one isoform and only one read from another isoform, and then you try to calculate your precision and recall, and then you may have a bias because, for example, you have a high coverage for very simple isoform, which is successfully assigned, and you have low coverage for complex isoform, vice versa. In any case, it may bias your precision and recalls. But when you have absolutely uniform coverage across the entire transcriptome, then you have no biases. You just estimate how well you deal with this type of transcriptome. So we compared ourselves against Scanty. Yes, Scanty is not meant for analyzing raw reads. It's meant for analyzing collapsed transcripts, but as there is nothing else which can, there are no other tools which can assign transcript sequences to known annotation, we had to compare ourselves against something. And as you see, for transcripts, for reference transcripts, and for PacBio data with lower rate, Anti and IsoQuant, they both do nearly perfectly, especially regarding precision, and I would say decent in terms of recall. But when it comes to nanopore data, you see that precision drops, but not significantly, especially for IsoQuant, but recall drops a lot. What is also important is simulated mouse data with trans-nano-SIM, so it's more realistic, and human data was simulated with ISO-seq-SIM, so it has uniform error distribution. Both have 11% error rate. What you can see here is simply error rate is not what important. You have 11% error rate in both data sets. The first one, the mouse one, has like nanopore specific errors and biases. So you see that both precision and recall is significantly lower for mouse data set, which means that not only purely error rate is important, but the type of errors is also what makes it hard to work with such reads. And the second experiment we carried out is assessed how well we restore transcript models. What we did here, we simulated reads again using the entire transcriptome from the entire annotation, and fed it into different transcript model construction tools, for example, into our tool and other tools I will list in the next slides. But we also provided them incomplete gene annotation. So we simulated reads from the entire transcriptome, then we removed randomly about like 5% of spliced isoforms from our annotation and gave those tools incomplete annotation. So from the tools point of view, you have some novel isoforms in your reads which are not present in your database. And then we compared what tools output. They all output GFF files, and we used GFF compare to make a comparison against what do tools produced against the original annotation and see how well they can reconstruct transcripts which were thrown away from the annotation but were present in the simulated reads. And of course, GFF compare allows to calculate sensitivity and precision as well. We did this for PacBio reads with low error rate. We did it for non-true nanopore data with uniform error rate, and as you see, all tools that we compared against, Tai2, Flare, Bamboo, they have pretty high precision in all cases, and I would say sensitivity. Well, maybe except Bamboo has like high sensitivity, but I'll mention that a bit later. But when it comes to real nanopore simulated data, like not real, but realistic nanopore data, you see that both precision and recall drop significantly, especially for string times there. For Bumble and IsoQuant, you see precision is pretty high, and we are proud that we have a bit higher precision, but we have significantly lower sensitivity. And why is that? The thing is, Bumble uses different strategy in comparison to all other three tools. StringTie2, Flare, and IsoQuant, they try to report a transcript only if they see a confirmation that this transcript appears within reads. Bumble always outputs complete input annotation, and in addition to this input annotation, outputs some novel transcripts. So even if some transcript is present in your annotation but is not present in the read, Bumble will output it anyway. So that's the difference. That's why it's not correct to compare sensitivity of Bumble to other tools, because other tools ignore isoforms which are not confirmed by any of the reads, or I don't know, confirmed by very few reads. In any case, Bumble does pretty well, and we consider that in terms of precision, we are doing pretty well too. And sensitivity is something we will also look into. But here, we also think that precision probably is a bit more important. It's better to have correct transcript models in your output than when you have all transcript models, but you also have like a lot of incorrect ones. So for example, like Flare and StringTie, they have precision like 66, 32. So it means they output a lot of trash transcripts, which is not probably good for your further research when you have false positive calls in your transcript models. We also decided to check is how well do those tools agree with each other, how consistent is their output. It appears that only 10% of like all union of all transcripts belongs to all three tools, which is not a lot. What we are proud of is that we have no transcripts in our output which are not supported by any other tool. We don't have unique hits. And on simulated data, as you see, like looking at this table and this graph, we can say that we don't have false positives a lot in comparison to string times there. Another point, of course, simulated data is good, and you can check different like precision recalls and so on and so on. But of course, we wanted to test ourselves on real data somehow. We used the real PacBio CCS and nanopore data publicly available. And as you see, for PacBio CCS, as you expect, much more reads can be uniquely assigned to known isoforms, 70% versus like 50%. For PacBio reads, you also have a small amount of ambiguous reads because you don't have much truncation as nanopore reads, like where you have almost 24% of reads that due to truncation were not mapped unambiguously. And of course, for nanopore data, you have more inconsistencies, like more than 50%. Of course, since you have more inconsistencies in your matching, you also find more novel transcripts. Of course, some of them may not be true novel transcripts, but some kind of artifacts that we missed. But in any case, for PacBio data, you have a few number of them. And again, what we could do, of course, we couldn't calculate precision and recall here because we don't know what is contained in the original sample. So we compared how well the tools are consistent with each other, and it appears that on real data they are even less consistent. You have for PacBio data only 8% of all transcripts that are common for all three tools, and only 7.3% for nanopore data. And again, as a positive point for us, we see that for PacBio data we generate only 0.1% transcripts which are not supported by other tools, and a bit higher number for nanopore data, 3.7. Still, it's much smaller number than transcripts supported by other tools. Okay, probably time for conclusions and question and discussions. We developed a new tool. It's already has been released, and we have high precision and I would say decent recall for different types of long-read sequencing data. We still think that analyzing nanopore data is a big challenge due to truncation, due to high error rate, but it's cheaper. I think we should invest time into analyzing nanopore RNA sequencing data. And what we also see as a conclusion that tools for transcript model construction are not very well consistent with each other, and we all have to improve on that. I'd like to thank all my team, and if you are interested in the tool, here's a GitHub link, and of course you are welcome to ask any questions now or later on emails or using GitHub issues or whatever. Thank you very much, and yeah, I'm ready for discussion.
Thank you very much, Andrey. Again, everyone, you can comment or go to the Isoforms channel. We will give some time just in case people have questions, because we do have time for questions and answers. Oh, it's
not expect that I have hours, because my previous talks were like about an hour long or so.
Okay.
And yes, I think I can share the slides, so you can go over the slides again anytime you want.
I see someone is typing. Okay, it seems that there are not no more questions. So thank you very much, Andrey, for sharing with us about IsoQuant. It seems like a nice approach to handle this type of difficult data. So we appreciate your time to explain this to us today and to answer our questions.
Yeah, thank you very much for inviting, and yeah, don't hesitate to write me if you have any questions later or any issues regarding the tool, if you will run it at some point.
Okay, thank you. Again, I want to remind you that all of our seminars are recorded and you can watch it again on YouTube. And I do have two short announcements before we end the seminar. First of all, a reminder that we have a virtual NGS symposium next month. So the abstract deadline is next week, 27th of August, and we welcome everyone to participate. And please remember to give us feedback on Discord. There's one question actually from YouTube. It's on the Isoform channel. Andrey.
Also a good question. Yeah, we don't do we don't do anything with internal priming yet. So like for bio, it's probably easier to handle it with IsoSeq. I'm not sure. I think it should do something with that, but currently, yeah, we don't have anything. And you have examples probably where you have like strong evidence of internal priming, and would greatly appreciate that, so we could check what's going on inside our tool when we have internal primer and how a liner behaves it, because yeah, as I said, we haven't studied this particular issue a lot.
Okay, it
But yeah,
seems that
the general answer is no. Yeah, we don't.
Yeah, the person asking the question said thank you also. So, and I don't see any more questions. So yeah, thank you very much. And the feedback form is already on Discord. And have a nice day, everyone. Thank you.
Thank you. Bye-bye.
Bye.